Yeah. So I'll be talking about the structural and functional determinants inferred from deep mutational scanning. Yeah. Mhm. Yeah. Yeah, sorry, I just got lost a little in the other screen. Yeah, so I'm back. So I'll be talking about the structural and functional determinants inferred from deep mutational scans. I did my PhD under Professor Raghavan Varadarajan at Molecular Biophysics Unit in Indian Institute of Science. So the work that I'll be describing today is the work which I did in my PhD. Currently, I'm co-mentored by Professor James Fraser and Willow Coito Mestres at UCSF, where I am studying, I'm making recombinatorial libraries of fusion oncoproteins and then screening them for their oncogenesis. So as we all know, deep mutational scanning is based on the fundamental notion of the ability of proteins to exhibit sequence structure function paradigm. It boils down to the most basic concept of molecular biology, where DNA is transcribed to RNA, which is further translated into an amino acid sequence. The primary structure of the protein dictates its tertiary structure, which folds into its quaternary structure, attains its functional form for the proteins to exhibit its function. So these deep sequencing techniques are very cleverly linked to some screen, which could be reporter based, which helps to identify the activity of each mutant. It has varied applications. It can be used to identify thermodynamically stabilizing mutations, or identification of active site and buried positions, or identification of um, mechanism altering mutations, or it also facilitates the prediction of protein secondary structure, or it also helps to distinguish strong binders from weak binders. The system that I worked on was a type 2 CcdA toxin-antitoxin operon, where both the toxin and the antitoxin components are part of the same mRNA and they are transcribed from the CcdA operon. If the toxin and antitoxin form a tight complex, it binds tightly to the operator promoter region, repressing the transcription of CcdA operon. However, under stress conditions, CcdA being labile is degraded, and the CcDB is now free to bind to its cellular target, which is DNA kinase, to exhibit its cell toxicity. It also forms a de-repressing complex, which results in fresh synthesis of CcdA CcDB operon, and it increases the transcription of the CcD operon. We wanted to develop a fissile method to deduce the entire structure of the protein, in our case, the toxin component of the toxin-antitoxin system, solely from mutational data, for which we generated a comprehensive site saturation mutagenesis library of the CcDB in CcdA, CcDB operon. We transformed them into a strain which is resistant to the toxic activity of the toxin. So here, all variants, both active as well as inactive variants of CcDB shall grow. The library is further transformed into a strain which is sensitive to the toxic activity of the toxin. So here, only now those variants will grow which have higher toxin levels than the antitoxin. Um, Only those cells will grow which have lower toxin levels than the antitoxin, while the variants which have higher toxin levels than the antitoxin will die. Further, the mutants were deep sequenced, and we assigned a variant score in the form of fitness scores. These are fitness scores, which are known as relative fitness CcDB scores, and we plotted the distribution of all the mutants for these variant scores. We found and we overlaid the distribution with the synonymous mutant distribution. We see, which acted as an internal positive control. We see that most of the synonymous mutations, as expected, are enriched in the region which is in twofold range of the wild type. We use these to assign our cutoffs to describe which mutants will be assigned as an hyperactive phenotype, whereas which mutants will be assigned an inactive phenotype. So when I say hyperactive means the mutants which are able to display a toxic activity higher than that of the wild type. And when I say the inactive mutants, these are the mutants which are able to display their activity lower than the wild type. We further categorize these mutations into four structural categories. One was CcdA interacting mutants. The other is the gyrase binding site mutants, buried site mutants, and the exposed non-active site mutants. We can clearly see that the CcdA interacting mutants, which is the antitoxin interacting mutants, show a distinct phenotype from the rest, where the majority of the mutants are concentrated on the hyperactive side. But we could not distinguish gyrase binding site from buried site as both displayed inactive phenotypes. And as expected, the exposed non-active site mutants displayed a phenotype similar to that of the wild type, basically a neutral phenotype. We further validated this assay where we have like constructed a subset of CcDB mutants and carried out a low throughput spotting assay. And we see that the mutants which have a low relative fitness CcDB score, like S12G, R13G, are the ones which are not surviving the sensitive strain, whereas the mutants which are like E87P, D19Y, which have a much higher relative fitness CcDB score than the wild type, are surviving higher, show cell growth propensities higher than the wild type. We had also constructed a few of these CcDB mutants and checked the soluble fraction. And we see that the CcDA interacting mutants have their solubility comparable to that of the wild type, while the mutants which displayed an inactive phenotype, maybe could be because of some degree of destabilization, showed lower fraction solubility in the protein. We now, to understand the molecular mechanism behind the observed phenotypic effects from the CcDB sensitive strain, we designed another assay where we have constructed the Rayleigh reporter gene downstream of the CcD promoter. And we played around with the Shine Dalgano sequence, and we saw that the one which has the consensus Shine Dalgano sequence gave us maximum expression for Rayleigh. We constructed this assay in a strain which is resistant to the toxic activity of the toxin in order to avoid any ambiguous effects of the toxicity from CcDB. So now we are able to screen the effects of CcDB mutants only by the toxic levels of Rayleigh. So here, this assay is a measure of how efficiently the CcdA CcDB forms a complex. If it forms a tight complex, it will repress Rayleigh expression and therefore decrease will increase cell growth. Whereas if it forms a de-repressing complex, it will increase Rayleigh expression and therefore we will observe more cell death. We further validated this assay by purifying a few CcDB mutants and then carrying out a thermal shift assay in the absence and in the presence of CcdA. So we clearly see that the mutants which had a higher relative fitness Rayleigh score, that means which show stronger binding to CcdA, also have a higher delta Tm value, which means they have a higher thermal shift than the wild type. And vice versa for the case where the mutants had a lower relative fitness Rayleigh score. So therefore, validating our Rayleigh reporter assay, we find that the two phenotypic readouts together help to unveil the molecular mechanisms that were obtained from the deep sequencing data. We observed that most of the CcD interacting residues are enriched in the hyperactive and de-repressing class because CcDB here will be impaired in binding to CcDA. Most of the gyrase binding site residues are enriched in the inactive and repressing class because here CcDB is impaired in binding to DNA gyrase. However, CcDA binding is retained. We observe that most of the most of the buried site residues are enriched in the inactive and de-repressing class because since CcDB will be misfolded, the binding to both CcDA as well as DNA gyrase is impeded. We wanted to now further identify critical CcD interacting residues from mutational data, since when we compare it with the structural data, we observed solely structural data will not suffice the will not suffice this issue. Structural data along with the mutational data helps to identify critical residues which are important in binding to CcdA. So we find that the mutant for the residues for which mutants are maximally enriched in the hyperactive and the derepressing class are the residues which are crucial for CcdA binding. We further mapped the residues which were part of the hyperactive and derepressing class onto the CcdA CcDB complex, and we see that majority of the CcD interacting site residues are part of the true positives, therefore validating our assay. We further wanted to do biophysical studies and validate our inferred mechanisms. So we took a CcDA interacting residue, which is CcDA interacting mutant, which is R13G, and carried out assays to find out whether it's binding to CcDA or not. We carried out thermal shift assay as well as microscale thermophoresis, and both of them clearly show that R13G uh, is no longer binding to CcDA, while the wild type still retains its binding to CcDA. We also further found out that the residues which are important in binding to CcDA are also important in rejuvenating CcDB from the CcDB gyrase complex, for which we designed an assay where we have immobilized gyrase on a CM5 chip. We pass a fixed concentration of CcDB over DNA gyrase, allowing it to bind to DNA gyrase. We further pass increasing concentrations of the CcDA. We use a CcDA peptide, which is the C-terminal region, which is the region which is actually involved in binding to CcDB. So if here the CcDB is able to bind to CcDA, which is the case for wild type, it will happily rejuvenate CcDB and dissociate CcDB from the CcDB DNA gyrase complex. While if there is a mutation which is defective in binding to CcDA, it will no longer be able to rejuvenate or dissociate CcDB from the CcDB gyrase complex. And this is exactly what we observe, that for the R13G mutation, even at the highest CcDA concentration that we pass through, we see there is four-fold lower dissociation compared to that of the wild type. We further wanted to identify important important critical residues for gyrase binding site. And we see that here, majority of the gyrase binding site residues, those show inactive and repressing phenotype, but still critically, when we compare the structural data with the mutational data, we find that the residues 87 and 100, so the mutations are mostly enriched at these residues, and these seem to be the ones which are critical in binding to DNA gyrase. We further validated by doing an SPR study where we have done a binding study of the E87P, a gyrase binding site mutant, and we see that there is almost no binding compared to that of the wild type. We further evaluated the performance of our method, and we see on an average 70%, our method is able to discriminate between multiple binding partners and distinguish them from buried site mutants and the exposed non-active site mutants with around 70% accuracy. So as in one of the slides shown before, we saw that majority of the buried site mutants were giving a neutral phenotype. So this is a very interesting result that caught our attention, because it is contrary to what is generally expected, since we expect that the buried site mutations will be destabilizing and therefore they will have an inactive phenotype. So, and this is what we have observed in a previous assay where we have plated the CcDB site saturation mutagenesis library, which is now in the absence of CcDA at increasing inducer concentrations. And so the mutants which are growing at the highest inducer concentration are the ones which are conferring a destabilization to the destabilization effect to the CcDB protein. And these are generally core mutations. So the mutational score that we had assigned for this assay was the mutational sensitivity score, which implies a higher mutational sensitivity score is equivalent to more destabilization and therefore higher cell growth for those CcDB mutants. In the present work, we have estimated the CcdA binding through this Rayleigh reporter assay, where the higher relative fitness Rayleigh score implies that CcDB is retaining its fold and is able to bind to CcdA efficiently, therefore repressing the Rayleigh expression, and therefore there's more cell growth. So we further compared the deep sequencing data of these two assays, and we observe that in the absence of CcdA, most of the buried site mutations to charged and polar residues are inactive, and this is what is expected. But very interestingly, on the contrary, we see that most of these CcDB mutants regain back their fold in Operonic context when they are present in the presence of CcdA. Uh, so to understand whether it is a post-translational or a co-translational effect, we further made a few of the CcDB buried mutants in Operonic context. And also, so that means when both the CcD and CcDB components are expressed from the same mRNA versus when the CcD and CcDB are expressed from different mRNAs. And then we did this in vivo solubility assay to understand the fraction solubility of these mutants in supernatant versus pellet. And we can clearly see that when the CcDB buried site mutants are expressed from different mRNAs, most of those mutations are being destabilized and are directed to inclusion bodies. Whereas in the case of Operon, they are mostly present in the supernatant. That means the fold is being regained and they are forming their native CcDB fold. We further purified these mutations in Operonic context and observed and estimated the relative protein yield of these mutants relative to the wild type. And we see that for some of these CcDB mutants, there is even like 1.5-fold higher protein yield than the wild type. We further carried out some structural analysis on the CcdA-CcDB complex, and we see that there's a large interface which is formed at the overlapping C termini end of the CcdA monomers, which is interacting with CcDB. We speculate that the intrinsically disordered region of which is the C-terminal tail of the CcdA interacts with and stabilizes the CcDB nascent chain prior to homodimerization. And this possibly prevents its erroneous association with other proteins in a crowded cellular environment, which includes other toxins and antitoxins. And this also prevents the CcDB binding to its cellular target, DNA gyrase. So our study emphasizes on the fact that CcdA CcDB forms a co-translational assembly. It emphasizes on the fact that they form a complex early on in the translation, and they are part of co-translational assembly, which in turn helps the CcDA to relieve any folding defect in CcDB, and therefore prevents their association with DNA gyrase or their erroneous association with any other cellular components, resulting in formation of tight CcdA-CcDB complex in the cell. So when we saw the site saturation mutagenesis library, a very interesting observation caught our eye that contrary to what is expected, we found that some of the synonymous mutations were displaying a very distinct phenotypic effect than what is expected from the wild type. So we now generated a single synonymous codon library in the background of the CcD operon to estimate the effects of these synonymous mutations. This was done via inverse PCR methodology. We further validated the assay, and we can clearly see for the mutants which had a lower relative fitness CcDB are the ones which are not surviving in the sensitive strain, versus the ones which had a higher relative fitness CcDB score are the ones which are surviving and are growing better than the wild type. We further averaged out the mutational scores for all the synonymous mutations for each position, since we wanted to look at the positional effects for these mutants. we find that synonymous mutations in the N-terminal region are the ones which display large diverse phenotypic effects. So it implies that the initiation phase is the major contributing factor for the to translation efficiency, which is followed in order with by the elongation phase and the termination phase. We further wanted to understand the impact of codon usage on protein abundance, for which we have calculated the moving average of relative codon usage over a sliding window of 5 mutants. And similarly, it was done for our variance score, which is a proxy of the effects of CcDB toxicity. And we can clearly see that mutations to rarer codons are increasing CcDB abundance in cells. So this is observed that in general, in naturally occurring genes, rarer codons are enriched in the N-terminal region, which increases the expression of the protein. In our case, likely since it's the toxin, it means that the wild type gene has the preferred codons, since mutations to those preferred codons are the ones which are increasing the translation initiation. And they have been evolved to have those preferred codons in order to reduce CcDB abundance, which in turn will help the cell to survive. We further carried out some correlation studies and we found very interesting results where we find opposite trends in the N-termini and the C-terminal region. We see that the mutations to rarer codons increase translation efficiency in the N-terminal region. On the other hand, mutations to preferred codons increase translational efficiency of the gene in the C-terminal region. And when we did some correlations with the GC content and the mean free energy, that is these a proxy of how stable the mRNA structure is, it suggested that the stable mRNA structures reduce translation initiation. And this could be because if there is a higher, if the mRNA structure is very stable, it might occlude the Shine-Dalgano sequence, preventing translation initiation. So therefore, it is not just codon rarity, but also reduced mRNA structure that increase translation initiation. We further plotted the relative fitness CcDB scores for the mutants that belong to the hyperactive derepressing class, which implies that these are the mutants which are involved in enhancing the translational efficiency of CcDB. We observe that the mutations in the N-terminal stretch, which is from 5 to 13, are the ones which exhibit maximum fold increase in translation efficiency. Very interestingly, we see that altering synonymous codon usage at a single site in the N-terminus resulted in 100-fold increase in translation efficiency, even though 99% of the gene remained same. So this intrigued us, and we wanted to now generate an entire synonymous mutant library, but in background of a parent hyperactive mutation, which is an N-terminus hyperactive mutation. So we created an N-terminus hyperactive mutation, and we studied the effects in that background for all the other mutations. So first, we validated that assay, and we could see very diverse effects, where we can clearly see that the mutants which are showing a very low relative fitness CcDB scores are the ones which are not surviving in the sensitive strain, while the mutants which have a higher relative fitness CcDB scores are the ones which are surviving, which are growing more than the wild type. We further wanted to look at the positional effects, and very interestingly, we observed that presence of a hyperactive mutation in the N-terminal region increased translation efficiency sufficiently such that now the effects in the elongation phase and the termination phase became more apparent. And we see that around 82% of the mutants exhibited a highly toxic phenotype. That means a hyperactive phenotype in the CcDB sensitive strain, while giving a repressing phenotype in the Rayleigh reporter strain, implying that this effect can be only observed due to alteration in folding kinetics. So if you see, we have these specific stretches, which is 52 to 57 and 72 to 77, which are giving a more distinct phenotype than the rest of the mutants. We wanted to now understand why is it that these specific regions are giving a more distinct phenotype. We did some correlation studies to understand the likelihood of the presence of translational pause sites at these sites, and we in these regions, and we found that wild type codons introduced translational pause sites, but due to synonymous mutations, these translational pause sites were abrogated. And this suggests that abrogation of translational pause sites in slow folding elements like beta fold, beta sheet, and a loop region with a short helical turn, um, due to synonymous mutations, resulted in misfolding of the protein. We further compared the mutational scores for these synonymous mutations with the structural properties like the hydropathy index, and we saw that there's a pattern where we see that the stretches 52 to 57 and 72 to 77, which have a more inactive phenotype than the rest of the mutations, had a higher hydropathy index, that means lied in the hydrophobic regions, implying that hydrophobic regions seem to have spatial translation kinetic requirements that ensure proper folding of the protein. This implies that due to synonymous mutations in the hydrophobic regions, uh, there is misfolding of the, I mean, there's misfolding of the protein, and because of which they get exposed to the solvent region, therefore resulting in misfolding of the protein. So the insights gathered on synonymous mutations from our study can be used to optimize heterologous gene expression in E. coli, and also helps in understanding the molecular basis of alteration in gene expression. In summary, we use deep mutational scanning to develop a fissile method to deduce the entire structure of the toxin, which has multiple binding partners in its Operonic context. This work is published. Um, then we also found out that CcdA is able to relieve folding defect, an inherent folding defect in CcDB, which implies or suggests that CcdA chaperones CcDB against irreversible misfolding and aggregation via co-translational folding mechanisms. So this work will be soon out in bioRxiv. And the other very interesting phenomena we saw that contrary to what is expected, single synonymous mutations, which are expected to exhibit minimal or no effects, we are seeing much diverse and distinct effects than the wild type. And we wanted to further understand the molecular basis of strong phenotypic effects of such single synonymous codon substitutions in the toxin gene. And this work is also published. So please feel free to check out these works if you're interested in understanding how we can use deep mutational scanning for varied methods, and we can address some very fundamental questions in molecular biology using DMS based on which can be related to protein structure activity, stability, and co-translational folding mechanisms or protein folding. Yeah. Thank you. And I would like to acknowledge my professor, Professor Raghavan Varatharajan, under whose guidance I did this work. He is the first one who introduced me to this powerful technique in this arena of deep mutational scanning. I also would like to thank all the past and present members of RV Lab, Ujjwalji, Sameer, Kritika, who helped me out were initial mentors during my learnings during my graduate studies, specifically my collaborators, Pihu, Munmun, and Kavya. Pihu was part of the co-translational folding project. Munmun has done some computational analysis in the synonymous mutant project, and Kavya has processed the deep sequencing data for the site saturation mutagenesis library. So thanks to all of them for because of whom we could carry out this work, and it was possible to be out in the public domain, and it's for everyone to check out. And yeah, if there are any questions related to deep mutational scanning or and I'd be happy to collaborate if there's anything that anybody would want to know about it. So yeah, thank you.
Thank you, Priyanka, for the really nice talk. Okay. Meanwhile, maybe the people will write their question in the Q&A. I have like one or two questions that I'm mainly a computational person, so like the first question is like you made a lot of assumption around the fact that the protein like can lose this folding and everything, especially in the hydrophobic core. Did you ever like try any in silico tool that can support that? That can support the fact that, that you can have this losing the complex itself.
Um, yeah, so we have done some studies where we have checked for the solubility assays. Um, but I think because of the synonymous mutations, it is more hard to find out by in vitro experiments since they are not able to mimic exactly what is happening in vivo. So it is more important to devise more high throughput ways to maybe validate or cross confirm what is observed from these in vivo activity assays, specifically in case of synonymous mutations. For the non-synonymous mutations, it's much more easier. So we have done a bunch of biophysical studies to validate their effects.
Yeah. And since there is nothing yet, I'll ask the second. So at a certain point, you talk about the C term of CcDA
Mm.
interfering with the formation of the complex, right? Is
Yeah.
this, sorry, at the end I didn't capture, is this like, is like a self-interaction of the CC terminal in the on the protein that prevents the complex if there is a mutation, or is still like a co-translational like mechanism that is behind this?
So we speculate it's a co-translational mechanism. Specifically, our data supports that since we have constructed these constructs in Operonic versus in non-Operonic context. So there are studies which show that even if, let's say, CcDA is made in proximity with CcDB, it should be able to relieve the folding defect, but that is not what we observe. Um, we observe it only when it is part of the same polycystonic mRNA. So that is like the biggest evidence which shows that CcDA binds to CcDB prior to its homodimerization. And there are many different structures of CcDB dimers and CcDA dimers which support our inference that it is that CcDA forms association with CcDB early on in translation, early on, like before it is fully translated and then they form a complex. So it is early on during the translation phase that they form a complex, which helps in the proper folding of the CcDB. And therefore, maybe they are the gene organization is also such that they have evolved to form to be part of the operon such that first CcDA gets synthesized and then CcDB, which will in turn help in binding to CcDB because the C-terminal region of CcD is intrinsically disordered, and that gets ordered only upon binding to CcDB.